This is a real human skeleton. What you're looking at might seem more appropriate for a low-budget 80s sci-fi horror, or an avant-garde art exhibit, than the skeleton of an actual human body. But reality is often stranger than fiction. This is the real human skeleton of Harry Raymond Eastlack, donated to the Mutter Museum in Philadelphia after he passed away. Eastlack is a victim of an incredibly rare disease known as FOP, Fibrodysplasia ossificans progressiva, a genetic condition where one's soft tissues progressively ossify. They turn into bone. The deformations you see in Eastlack's skeleton are what happens when a person's skeletal muscles and connective tissue undergo a lifetime of ossification, transforming their free-moving skeleton into an immobile cage. This sort of thing, by all means, should be an isolated case, the outcome of an incredibly unlucky series of defects. After all, only a true disaster can produce something as mangled as this. Your body should know that flesh is flesh and bone is bone. But anyone who looks into this disease, even a little, will uncover something disturbing. This condition, this aberration of the human body, is only caused by a single mutation in a single gene. Your skeleton is just one genetic accident away from looking like this. So, how can this happen? Let's start with a bit of context. FOP is an insanely rare disease, but it's obviously not just isolated to Eastlack, with an incidence of about 1 in 2 million people. With enough cases of FOP, we've actually managed to outline the progression of this condition, to the point of identifying the gene responsible for it back in 2006. It's what we know about this physical progression that's actually going to tell us a lot about the genetic mechanism. And so, that's where we're going to start. At birth, FOP patients don't actually show any substantial defects, with the possible exception of some minor bone deformations, specifically bent great toes. This trait, caused by the fusion of the phalanges and an overgrown metatarsal, is pretty expected for a condition that creates excess bone. But what's unexpected is how there's little else at play. Young children with FOP will seem fine, at most showing signs of stiffness around the neck. But FOP's progression is gradual, it's slow and insidious, and only at about 5 years of age will the average FOP patient expect their first flare-up. Flare-ups are events where parts of an FOP patient's body swell up, accompanied by intense pain. Flare-ups will occur episodically throughout a patient's life, but it's important to note that these events have patterns. Flare-ups that happen spontaneously as the patients grow older will first start in the proximal axial regions of your body: the neck, the spinal column, and the thoracic ribcage. Only later in life does bone formation begin distally, outwards towards your hips, knees, and wrists, locking the joints. A more unusual pattern can be observed when flare-ups happen non-spontaneously. FOP can trigger as a response to injury, where wounds to an FOP patient's soft tissue, such as the skeletal muscle and tendons, will result in that area getting ossified. This bit is especially devastating when you consider what actually constitutes injury. Accidental wounds certainly count, but so do more intentional ones: vaccinations, injections, surgical incisions. FOP, being a rare condition, is often misdiagnosed, specifically for tumors. And when physicians attempt to surgically collect a sample for biopsy, it can often worsen the condition by initiating a new flare-up. It's an intensely cruel condition, especially as newly created bone begins to fuse with old and new bones. Over time, a patient will become immobile as their axial skeleton, shoulders, and joints lock in place. It's a slow, suffocating fate that often ends in premature death, with a life expectancy of just about 40 years, as a disfigured and inflexible ribcage restricts the volume of the patient's lungs and their ability to breathe. This thoracic insufficiency syndrome paves the way for fatal respiratory problems, like the pneumonia that Eastlack died from 6 days before his 40th birthday. FOP is terrifying, but it's also strangely predictable. In FOP, flesh is not transformed into bone at random, nor is it a runaway process. It's nearly undetectable at birth, happens episodically and progressively during growth, and consistently occurs in response to injury. There's a special mechanism at work here, a mechanism that seems to be caused by a malfunction in typical bone formation, activating the pathway for typical osteogenesis, but in the wrong places and at the wrong times. And so, at the core of FOP is a question not only about abnormal bone growth, but normal bone growth. If one mutation can cause a body to grow bone in the wrong places, what does bone growth in the right places look like? How does your body know what bone is? Like, seriously, how does it know? You can fully grasp the concept of a bone, what it looks like and where it should be, but your body isn't conscious like that. It needs some sort of mechanism to know where and what bone should be. And that mechanism starts with a little thing called stem cells. Stem cells are cells that are capable of becoming different types of cells, and without them, your body wouldn't exist. When egg and sperm first meet, only one cell is made, and if that cell wasn't able to become all the different types of cells in your body, you wouldn't exist. The zygote, the first cell in reproductive development, is totipotent. That means it's capable of becoming every cell in your body, from acid-secreting stomach cells to infection-stopping immune cells to, of course, bone cells. The act of deciding what type of cell a stem cell should become is called differentiation, and it's the process of controlling differentiation that decides whether bone should be bone. Now, to take this idea one step further, consider this. If every cell in your body originated from one totipotent stem cell, there should be something that all cells in your body have in common. And, well, there is. It's their DNA. Every cell in your body, with very few exceptions, have the same DNA passed down from their stem cell progenitors. This might seem counterintuitive, because you may know DNA as the blueprint for cell function. Surely, cells with different functions have different DNA. But if DNA is a blueprint that cells read to commit certain functions, stomach cells and bone cells don't need to have different blueprints to work differently. They just need to read different parts of the blueprint. Differentiation works not by changing the DNA of the stem cell, but by forcing the stem cell to express different parts of the DNA. This radically simplifies the decision-making process of differentiation. All the instructions to become bone are predetermined in a stem cell. It just needs a signal to tell it which instructions to read. There are many signals that play a role in deciding whether bone is bone, but one that is crucial to understanding FOP is BMP, bone morphogenetic protein. Let's break this name down. Proteins are molecules consisting of long chains of smaller molecules called amino acids. When we say DNA acts as a blueprint for a cell's function, we usually mean that it acts as a blueprint for its proteins. When DNA is expressed, depending on its signal, a protein is created with a different amino acid sequence. The arrangements of amino acids in a protein determines its structure, which, and this is important, determines its function. A protein's amino acid sequence determines its structure, which determines a protein's function. Bone morphogenetic, on the other hand, is very straightforward. It morphs bones genetically. It's the signal that causes stem cells, specifically mesenchymal stem cells, to express the genes necessary to become bone. The general mechanism by which BMP sends the signal is by binding to a receptor on the surface of a mesenchymal stem cell, which causes some structural change to occur. One very common process that occurs in signaling pathways like these is something called phosphorylation, the addition of a bulky, negatively charged molecule to a protein. Remember, the structure of a protein determines its function, and so phosphorylation is an easy way to change a protein's function. Once it has bound BMP, the receptor can phosphorylate a group of proteins called SMADs. The name SMAD is largely unimportant, but the act of phosphorylating SMADs lets them enter the cell's nucleus and bind DNA, where they can help the cell choose the instructions needed to become bone. Through this pathway, BMP acts as a signal for differentiation. It's with this pathway that not only allows the body to know what bone is, but where bone is, because BMP is a signal from outside the cell. A stem cell population separated from a local BMP source cannot receive the signal to differentiate, and thus the localization of BMP determines the localization of bone. For a normal body, bone formation is anchored to areas of BMP expression, shaping a healthy skeleton through a robust biological system. A system so fragile that one mutation turns this into this. It's finally time to introduce the culprits behind FOP, the first of which is the actual stem cell population behind the abnormal bone growth. Within soft muscle tissue, there actually exists a mesenchymal stem cell population, but it has a completely different function to typical mesenchyme. Fibroadipogenic progenitors, abbreviated FAPs, don't differentiate into bone cells, partly because of the system we've established earlier. In skeletal muscle, they're not exposed to BMPs. They mainly play a support role in helping muscles regenerate after injury. But they are still mesenchymal stem cells. They have the instructions to become bone, they just need the signal for it. And that's where our second culprit, the actual FOP mutation, finally reveals its trick. The mutation most famous for causing FOP is a single amino acid mutation in ACVR1, a type 1 BMP receptor. As we established before, the amino acid sequence of a protein determines its structure, which determines its function. This mutation, R206H, is the replacement of a single amino acid. And with this one change, the entire system of regulated differentiation falls apart. And thanks to decades of research, we know how. ACVR1 is a receptor that can be separated into three crucial domains: an extracellular binding domain, where BMP is actually bound, an intracellular kinase domain, where the receptor actually phosphorylates SMAD, relaying the signal. And the third one is what is described as a glycine serine domain, a structure that overlaps the kinase's active site. We'll call it the GS domain for the sake of brevity. It's this domain that FOP's most well-known mutation is found. By blocking the kinase, the GS domain works as a guard, physically preventing SMADs from accessing the active site properly. The position of the GS domain decides whether a signal can be delivered or not. I quietly mentioned that ACVR1 is a type 1 receptor, and at the risk of adding more info to this already incredibly dense video, I need to mention there's a type 2 receptor. The type 2 receptor's job is to ensure that when a signal is ready to be delivered, the type 1 receptor is ready. Type 2 receptors can be separated into two domains. The first one is just another binding domain that also binds BMP, and the other is a kinase domain that seeks to phosphorylate ACVR1's GS domain. As BMP binds both of these receptors together, it functions to bring the two closer in a process called dimerization. The resulting two-protein complex, this dimer, serves two roles. One is to phosphorylate the GS domain, changing its structure so it no longer blocks ACVR1's kinase, letting ACVR1 phosphorylate SMAD. You could think of it like opening a door to let SMAD into the kinase. The other is to scaffold an even larger complex, where two of these dimers come together to form a cluster of two ACVR1 receptors and two type 2 receptors. The resulting 4-receptor complex is called a tetramer. This tetramer is known to play a role in ACVR function, as the stability and the structural changes brought on by its formation is necessary for proper signaling. The actual reasons why aren't well understood. But all you need to know is this tetramer functionally acts as the active conformation of ACVR1. By acting as a link between ACVR1 and a type 2 receptor, a bridge that connects the two together, BMP forms the dimer necessary to send the signal. You can already guess what a mutation in the GS domain of ACVR1 can do. By changing the structure of this domain, the mutation shifts the domain away from the kinase active site, making it more open to SMAD phosphorylation even in the absence of BMP. The door is already open. Now, ACVR1 is open to phosphorylate SMAD without an external signal, transforming FAP cells inside the muscles into bone. The body, without any way to prevent itself from sending a false signal, doesn't know that flesh is flesh and turns it into bone. This is the theory. This is the theory behind FOP. It's intuitive. Based on everything we've learned so far, it makes sense. And within the last decade, research has proven that this mechanism is wrong. It's sadly wrong. We're missing something really important. Remember at the beginning of this video, I told you that the physical progression of FOP will tell us a lot about its genetic mechanism. FOP is a condition that acts slowly and predictably, notably being incredibly benign in newborns and flaring up during injury. The theory would, if it were true, outright contradict this information. Specifically, this theory describes constitutive action, continuous activation of ACVR1. It implies that the reason FAPs turn into bone is because the mechanism that would stop them no longer works. But if that were true, why don't newborns that absolutely have these stem cells not show any symptoms? Why do flare-ups happen episodically instead of continuously? Why did they happen so slowly? Furthermore, what does this have anything to do with injury? BMP and ACVR1 have very little documented interactions with soft tissue injury, yet the most notable pattern seen in FOP patients is that soft tissue injury consistently prompts a flare-up. Finally, one big flaw with this theory is that an external signal like BMP is still necessary to form this essential tetramer formation that we saw earlier. This theory gets close, but it doesn't completely explain away the missing signal. We're going to need one last hint. And so, to round off this wild ride, let me break down one more name. ACVR1 stands for Activin A receptor type 1. I didn't lie to you when I said that ACVR1 is a BMP receptor, but when it was first discovered, it was noted for its ability to bind something called Activin A. Activin is a protein that is part of the transforming growth factor superfamily, alongside BMP. As a result, it also acts as a signaling molecule, but unlike BMP, it historically has very little to do with bone. It's notably very active in inflammation, a process where the body prepares to respond to injured cells, from mobilizing immune cells to fight off infection, to replacing damaged tissue. You would be right to assume that it can circulate to FAPs when the surrounding muscle is damaged. Activin A binds ACVR1 in a manner very similar to BMP, dimerizing ACVR1 and type 2 receptors, forming that stable complex. But stopping there. For a reason that, as far as I can tell, is still poorly understood, activin binding does not result in SMAD phosphorylation. In normal ACVR1, activin A cannot replace BMP as a bone growth signal. But in FOP, there's a difference. The difference being that the GS domain no longer blocks the ACVR1 kinase from the beginning. And so, when activin A naturally binds both ACVR1 and the type 2 receptor, bringing them close together and forming a stable dimer, once SMAD binds, there's nothing that stops it from easily being phosphorylated and sending a signal. Activin A, with this mutated ACVR1, can functionally replace BMP. And now, we can finally assemble the full picture. Whenever an FOP patient's body detects tissue damage, inflammation will occur, sending cells to the site of perceived injury that release activin A. Activin A brings the mutant ACVR1 and type 2 receptors of FAP stem cells together, dimerizing them. And due to a change in a single amino acid, they will falsely send a signal that expresses the genes necessary for differentiation. By incorrectly matching the signal for bone growth to a signal for inflammation, the body confuses injured flesh for bone. It will only do so at certain times. Healthy fetuses in particular don't undergo this type of inflammation, but inflammation will occur naturally and episodically over any growing person's life. It's an absolutely fascinating story of biology gone wrong, one that can be found in a world where bone is bone, flesh is flesh, and reality is stranger than fiction. Hi, so if you're still watching, you've had the patience to watch up to this point, and I just want to thank you so much for that. This has been another very fun project with a twist that I experienced myself in real life. While I was researching this, I figured out that some of the most famous papers on FOP are the outdated ones with the incorrect theory, and it was just an interesting process trying to piece together what the right explanation was. I want to try to make more of these in the future, more in-depth explanations into some biological anomalies that explain the actual mechanisms behind them. And if you're interested in stuff like that, I highly recommend you subscribe to see more projects like these. Once again, thank you so much if you stuck around this far. I really hope you've learned something interesting today. Take care. Bye.